right, so this is my original cell. We call this a parent cell, right? This cell has 1, 2, 3, 4 chromosomes, right? So we say the diploid number is 4. The way you write diploid in shorthand is just 2n. That's like the short way to write it. All right? These 2 chromosomes are homologous to each other. Okay? That means the red one came from the mother, the blue one came from the father. They are the same size and the same shape, and they have similar kinds of genes, but they're not exactly the same. Same thing with these 2. They're homologous to each other. Ready? Okay? So they're 4. All right? So this cell is in G1, so it's doing regular cell jobs, that kind of stuff. What comes after G1? S, right? What happens in S? Good, everything doubles. Right? So all the chromosomes replicate. Okay, what comes after S? G2, right? So if you have G2, the cell is growing, getting prepared for cell division. Okay, what comes after G2? Good. Now, what makes meiosis different is that there are 2 sets of division. There is meiosis 1 and meiosis 2. And you need to be specific about which one you're talking about whenever you refer to any of the stages. Okay, so let's start with meiosis 1. Okay, so the first step is called prophase 1. All right, so what happens in prophase 1, right? And it's called prophase 1 because it's prophase of meiosis 1, right? So the nucleus goes away, right? The centriole that was there before, right? It replicates, you have a second one, and the second one moves over here, right? So you got a centriole over here. Okay? Now, something else happens in prophase 1 that didn't happen in mitosis. And the homologous chromosomes, they get together. So they pair up. Okay? And then what happens is that something happens that's called crossing over. So what that means is that chromatids from homologous chromosomes will actually cross over like this and break off a piece of themselves and exchange portions. And notice, it has to be 2 different colors that cross over. So it's not, so remember these 2 are identical to each other, right? So if they cross over, it's not a big, no one cares, right? These 2 are identical to each other. So it has to be the chromatids that are not identical, right? So they're different colors, right? So remember, because your parents were not related, right? So these two chromatids, right, are not, the red and the blue are not identical to each other because they came from different parents. Okay? Crossing over can be a little segment like this, or it can be like a whole branch of your chromosome can cross over.
What dictates that?
That's a really good question. I'm not sure. I don't know if we know either. And it can happen more than once too. Okay? So that's crossing over, and that's the end of prophase. Okay, what comes after prophase? Good. So we have metaphase 1. Okay? So we have the fibers come out. Right. So they line up along the middle. But notice the way they line up, instead of lining up in a single line, they line up doubled up, like this. Okay? This is another way variation is introduced into the process because the way they line up, this set and this set is random. What that means, so they lined up here with the reds on this side and the blues on this side. Everyone see that? They can also line up this way, where you have like red here, blue here, red here, blue there. You understand what I'm saying? Right? And this doesn't look that big a deal because it's only like 2 sets, but imagine if this was humans and we had like 23 sets all along the middle. The way they line up is random, and it's called independent assortment. Right? So that's a big thing, and it happens during metaphase 1. Okay, so they line up in the middle. What comes after metaphase?
Anaphase.
Good. So you have anaphase 1. Okay, so what separates now is homologous chromosomes separate from each other. So this is anaphase 1. Okay? What comes after anaphase? Good. Telophase 1, right? That means new nuclei form. So this becomes a nucleus, and this becomes a nucleus. All right? What comes after telophase? Cytokinesis, right? So we got cytokinesis 1. So that means that this becomes a cell, and that becomes a cell. So after meiosis 1, okay, you have 2 cells now. And each cell only has 2 chromosomes. So you went from having 4 chromosomes per cell to having half that, or 2 chromosomes in each cell at the end of meiosis 1. Each cell is now, we say it's haploid, because it has half the number of chromosomes than it started with. Okay? So each cell is now considered haploid. We okay so far? Good. for cytokinesis 1? What do you think? This is meiosis 2, right? So we got prophase 2.
Prophase 2.
Okay? Now, what happens is that each of the two cells now goes through meiosis 2 at the same time. Okay? Which actually looks exactly like mitosis. If you remember mitosis, this part should be easy. Okay? So I'm going to start with this cell first, and then I'll do it with this cell. Okay? So, prophase 1. All right? So I'm sorry, we're in prophase 2. So what happens in prophase 2 is that the nucleus disappears again. Right? And the centriole that was there before, it replicates, and then the second one moves over here and makes fibers. Right? No more crossing over or anything because the homologous ones have separated, right? So we can't cross over anymore. Okay? What comes after prophase? Good. So you got metaphase 2. All right? So they line up along the middle now with the fibers make them line up. Okay? So now they're in a straight line again. Okay? What comes after metaphase? Okay, so we got anaphase 2. So now you have chromatids that separate from each other, just like mitosis. Okay, that's anaphase 2. What comes after anaphase? Telophase. So we got telophase 2. So then you get new nuclei being formed. So this becomes a nucleus, and that becomes a nucleus. After telophase comes what? Cytokinesis 2. So this becomes a cell, and that becomes a cell. These are called daughter cells, by the way, because they're the result of a division. The original cell was called a parent cell. All right, so I'm going to do the same thing over here. Okay, so this cell is going to go through prophase 2, so that means nucleus goes away. Right, you got the centriole that was over here, replicates, the second one moves over here. We got the fibers. What comes after prophase again?
Metaphase.
Right, metaphase 2, so it lines up in the middle. After metaphase? Anaphase 2, so chromatids separate during anaphase, right? What comes after anaphase? Telophase 2, right? So new nucleus here, new nucleus there. What comes after telophase? Cytokinesis 2. So this becomes a cell, and that becomes a cell. So at the end of all of meiosis, right, you have 1, 2, 3, 4 daughter cells total, right? Shh. And each one of them, if you look at the chromosomes in them, none of them are the same. Do you understand? Right? And this is what makes meiosis so different from mitosis. Because remember, mitosis, you're making exact copies. Right? In meiosis, you're making daughter cells that look completely different, and each daughter cell only has half the number of chromosomes as the original cell. Remember, the original cell had 4? Now each cell is haploid, they just have half. So each cell just has 2. 1, 2, 1, 2, 1, 2, 1, 2. And the 2 that are here are not the same as the 2 that are there, not the same as these other 2. This is why. And because of this, the coolest thing is that if you're a guy, every single sperm that you make is genetically unique, is genetically different from every other sperm. If you're a woman, every egg you make is genetically different from every other egg you make because of what happens during meiosis. It introduces genetic variation. All right? How are we doing? We okay?
Yes.
Mm-hmm.
Yeah. Can you do this on your own table now? Yeah? You're going to have to.
I forget.
Okay. All right. So I got